Signal transduction pathway. A series of steps in which a received signal is converted to a specific cellular response. Quorum sensing. Ability of populations of bacteria to communicate and coordinate their behavior via signaling molecules. Biofilm. An aggregation of bacterial cells adhered to a surface. Cell-cell recognition. Animal cells may communicate via direct contact between membrane-bound cell surface molecules. Local regulators. A secreted molecule that influences cells near where it is secreted. Growth factors. Compounds that stimulate nearby target cells to grow and divide. Paracrine signaling. A secreting cell acts on nearby target cells by discharging molecules of a local regulator into the extracellular fluid. Synaptic signaling. A nerve cell releases neurotransmitter molecules into a synapse, stimulating the target cell. Hormones. One of many types of secreted chemicals that are formed in specialized cells, travel in bodily fluids, and act on specific target cells in other parts of the body, changing their functioning. Long-distance signaling. Endocrine signaling. Specialized cells secrete hormones into the body fluids, often blood. Reception. Target cell's detection of a signaling molecule coming from outside the cell. A chemical signal is detected when the signaling molecule binds to a receptor protein, first stage of cell signaling. Transduction. Converts the signal to a form that can bring a specific cellular response, second stage of cell signaling. Response. Transduced signal triggers a specific cellular response, ensures that the activities occur in the right cells at the right time and in coordination with the activities of other cells, third stage of cell signaling. Ligand. A molecule that specifically binds to another molecule, often a larger one. G-protein coupled receptor. A signal receptor protein in the plasma membrane that responds to the binding of a signaling molecule by activating a G-protein, also called a G-protein linked receptor. Receptor tyrosine kinases, RTKs. Enzyme-linked receptor, ligand binding causes dimerization and autophosphorylation. Ion channel receptors. Binding of a ligand to the receptor results in an opening of a gate through the plasma membrane that allows entrance of the ions. The increased ion concentration in the cytoplasm either propagates signal transduction or results in a direct stimulation of a response. Protein kinases, an enzyme that transfers phosphate groups from ATP to a protein. Protein phosphatases, enzymes that can rapidly remove phosphate groups from proteins through dephosphorylation. Second messengers, a small, non-protein, water-soluble molecule or ion, such as calcium ion or cyclic AMP, that relays a signal to a cell's interior in response to a signal received by a signal receptor protein. Cyclic AMP, a ring-shaped molecule made from ATP that is a common intracellular signaling molecule. Second messenger, in eukaryotic cells, it is also a regulator of some bacterial operons. Adenylyl cyclase, an enzyme that converts ATP to cyclic AMP in response to an extracellular signal. Scaffolding proteins, a type of large relay protein to which several other relay proteins are simultaneously attached to increase the efficiency of signal transduction. Apoptosis, programmed cell death. Reaction center complex, a complex of proteins associated with a special pair of chlorophyll molecules and a primary electron acceptor, located centrally in a photosystem. This complex triggers the light reactions of photosynthesis. Light harvesting complex, a complex of proteins associated with pigment molecules, including chlorophyll A, chlorophyll B, and carotenoids, that captures light energy and transfers it to reaction center pigments in a photosystem. Primary electron acceptor, the acceptor of the electrons lost from chlorophyll is a molecule in the thylakoid membrane. Photosystem I, one of two light harvesting units of a chloroplast's thylakoid membrane. It uses the P700 reaction center chlorophyll. Photosystem II, one of two light harvesting units of a chloroplast's thylakoid membrane. It uses the P680 reaction center chlorophyll. Photorespiration, a metabolic pathway that consumes oxygen and ATP, releases carbon dioxide, and decreases photosynthetic output. Generally occurs on hot, dry, bright days, when stomata close and the oxygen concentration in the leaf exceeds that of carbon dioxide. Bundle sheath cells, a type of photosynthetic cell arranged into tightly packed sheaths around the veins of a leaf. G3P, A 3-carbon sugar, for each CO2 are formed in the Calvin cycle. 1 leaves to be used in the cell. 5 are used for regeneration of our UBP. Mitosis. In eukaryotic cells, a process of cell division that forms 2 new nuclei, each of which has the same number of chromosomes as parent cell. Meiosis. Genetics, cell division that produces reproductive cells in sexually reproducing organisms. Cytokinesis. Organic process consisting of the division of the cytoplasm of a cell following karyokinesis, bringing about this separation into 2 daughter cells. Chromatin. The complex of DNA and proteins that makes up a eukaryotic chromosome. When the cell is not dividing, it exists as a mass of very long, thin fibers that are not visible with a light. Sister chromatids. Replicated forms of a chromosome joined together by the centromere and eventually separated during mitosis or meiosis II. Centromere. A specialized condensed region of each chromosome that appears during mitosis where the chromatids are held together to form an X shape. Cell cycle. Series of events that cells go through as they grow and divide. Interphase. The period of the cell cycle during which the nucleus is not undergoing division, typically occurring between mitotic or meiotic divisions. Centrioles. Located near the nucleus and help to organize cell division. Nucleoli. Dense masses of RNA and protein that manufacture ribosomes. Several of these are located in the nucleus. Prophase. First and longest phase of mitosis, during which the chromosomes become visible and the centrioles separate and take up positions on the opposite sides of the nucleus. Metaphase. Second phase of mitosis, during which the chromosomes line up across the center of the cell. Anaphase. The third phase of mitosis, during which the chromosome pairs separate and move toward opposite poles. Telophase. The final stage of meiosis or mitosis, in which the separated chromosomes reach the opposite poles of the dividing cell and the nuclei of the daughter cells form around the 2 sets of chromosomes. Cell plate. A double membrane across the midline of a dividing plant cell, between which the new cell wall forms. during cytokinesis. Cleavage furrow. Pinching of the cell drawstring develops in animal cells only. Meiosis II. The second phase of meiosis, consisting of chromatids separating, along with the two diploid cells, splitting in two. Prophase I. The replicated chromosomes condense, homologous chromosomes pair up, crossing over occurs between homologous chromosomes, the spindle is formed, and the nuclear envelope breaks apart into vesicles. The longest phase of meiosis. Anaphase I. The third phase of meiosis, the replicated homologous chromosomes are separated, the tetrad is split and pulled to opposite sides of the cell. Metaphase I. The paired homologous chromosomes, tetrads, align at the center of the cell, the metaphase plate, in meiosis. Telophase I. The fourth of meiosis I. The number of chromosomes is now reduced by half. After this phase, the cell is considered to be haploid. Note, however, that the chromosomes are still replicated, and the sister chromatids must still be separated during meiosis II. Prophase II. The first phase of meiosis II, identical to the mitotic step, except that the number of chromosomes was reduced by half during meiosis I. Metaphase II. The second phase of meiosis II, identical to the mitotic step, except that the number of chromosomes was reduced by half during meiosis I. Anaphase II. The third phase of meiosis II, the sister chromatids are finally separated at their centromeres and pulled to opposite sides of each cell, is identical to mitotic anaphase, except the number of chromosomes was reduced by half during meiosis I. Telophase II. The fourth and final phase of meiosis II, the number of chromosomes was reduced by half during meiosis I. Haploid. Term used to refer to a cell that contains only a single set of chromosomes and therefore only a single set of genes. Diploid. Genetics. An organism or cell having two sets of chromosomes or twice the haploid number. Synapsis. The side-by-side pairing of homologous maternal and paternal chromosomes at the start of meiosis. Tetrads. The paired chromosomes consisting of 4 chromatids. crossing over. The interchange of sections between pairing homologous chromosomes during the prophase of meiosis. Somatic cells. Any cell other than a gamete has 46 chromosomes. Body cells. Gametes. Sex cells. Zygote. A fertilized egg. G1 checkpoint. Checks to see if cell size is adequate, chromosomes replication is successfully completed, and checks for DNA errors. G0 Phe. If conditions are not appropriate for the cell to divide, or if it is not programmed to divide, they are in this phase. G2 checkpoint. Asks if DNA replication has occurred, go-ahead signal triggers meiosis. Cyclin-dependent kinases. CDK enzymes activate proteins to regulate the cell. Growth factors. Factors that stimulate the cell to divide. Density-dependent inhibition. The phenomenon observed in normal animal cells that causes them to stop dividing when they come into contact with one another. Anchorage dependence. The requirement that to divide, a cell must be attached to a solid surface. Allele. One of two alternate forms of a gene that can have the same locus on homologous chromosomes and are responsible for alternative traits. Law of segregation. Mendel's law that states that the pairs of homologous chromosomes separate in meiosis so that only one chromosome from each pair is present in each gamete. Law of independent assortment states that allele pairs separate independently during the formation of gametes. Monohybrid cross. Hybridization using a single trait with two alleles, as in Mendel's experiments with garden peas. Dihybrid cross. Hybridization using two traits with two alleles each. Incomplete dominance. Creates a blended phenotype. One allele is not completely dominant over the other. Codominance. A condition in which both alleles for a gene are fully expressed. Multiple alleles. Three or more forms of a gene that code for a single trait, such as blood types. Apoptosis. A type of gene interaction in which one gene alters the phenotypic effects of another gene that is independently inherited. One gene masks the expression of a different gene for a different trait. pleiotropy. A single gene having multiple effects on an individual's phenotype, more than one phenotypic expression. Linked genes. Genes located on the same chromosome that tend to be inherited together in genetic crosses. Sex-linked inheritance. Traits located on the sex cells: X, color blindness, hemophilia. Homologous chromosomes. Chromosomes that have the same sequence of genes, that have the same structure, and that pair during meiosis. Autosomes. Non-sex chromosomes. P generation. Parental generation, the first two individuals that mate in a genetic cross. F1 generation. The first offspring from a cross of two varieties in the parental P generation. F2 generation. The second generation of offspring, obtained from an experimental cross of two organisms, the offspring of the F1 generation. Pedigree. A diagram that shows the occurrence of a genetic trait in several generations of a family. Tay-Sachs. Recessive lipid buildup causes fat buildup and brain death by 2. Sickle cell anemia. A human genetic disease of red blood cells caused by the substitution of a single amino acid in the hemoglobin protein. It is the most common inherited disease among African Americans. Cystic fibrosis. A human genetical disorder caused by a recessive allele for a chloride channel protein, characterized by an excessive secretion of mucus and consequent vulnerability to infection, fatal if untreated. 4%. Whites are carriers, most common lethal genetic disease. Huntington's disease. Genetic disorder that causes progressive deterioration of brain cells, caused by a dominant allele. Symptoms do not appear until the age of 30 or so. Albinism. Autosomal recessive, a hereditary condition characterized by a partial or total lack of melanin pigment, particularly in the eyes, skin, and hair. Camplants. Store the organic acids made at night in vacuoles and use them for photosynthesis during the day, when stomata are closed. C4 plants. A plant that changes CO2 into a 4-carbon compound before entering the Calvin cycle for photosynthesis. C3 plants. More than 95% of plants on the Earth are this, a plant that changes CO2 into a 3-carbon compound before entering the Calvin cycle for photosynthesis. MPF. Protein complex required for a cell to progress from late interphase to mitosis. The active form consists of cyclin and a protein kinase.